Today I'd like to talk to you about the work we're doing in my lab in Cambridge, working on the development of the brain. And in particular, we work on neural stem cells. And we're interested in how neural stem cells become quiescent, how they go to sleep, and most importantly, how to wake them up again. So I think many of you will be aware that the brain can be shaped by the environment, and this occurs at various stages of life. Not sure which one I can see best. During the development of the animal in utero, the development of the brain can be affected by maternal diet, immunity, and toxic load, and this can have effects on the structure and neurogenesis of the brain during development. But also later in life, there are stimuli that can affect the brain. So, for example, diet, exercise, disease, and injury. Now, we've learned over the last few years that there are neural stem cells not just but also in the adult. And it's known now that neural stem cells can generate neurons throughout development. And as I mentioned, they're important for, firstly, generating the first neurons in the brain in utero, but also later in life. They're important to generate neurons to maintain homeostasis in the brain and also for repair. Now, there's been a lot of interest in adult stem cells over the years, and what's particularly interesting is that adult stem cells in many different tissues in the body spend much of their time in a dormant state. They're not dividing, and we call this a quiescent state. And this is true not only in the brain, but also in other tissues and organs. So most of the stem cells are asleep. However, they can be woken up, they can be reactivated in order to generate new neurons when required. And the neural stem cells are able to respond to various different metabolic stimuli, and these include nutrition, exercise, and, as I mentioned before, injury or disease. And in response to these stimuli, they leave the quiescent state, they resume proliferation, and they generate differentiated progeny. Now, one of the reasons that we're so interested in this time of development and this process of going from quiescence to proliferation is that, as you can well imagine, if we had a way of waking up stem cells that are already in the brain, then it might be a way of inducing these stem cells to produce neurons so as to repair or replace damaged neurons after disease or injury. And this, I think, would be a preferable way to repair rather than doing transplantations into the brain. If we can stimulate the neural stem cells in situ, I think that might be a preferable way to repair the brain. So what we are very interested in is what regulates neural stem cell quiescence and reactivation. What allows these dormant cells to start proliferating again. Now, I've been talking about vertebrates so far, about human beings, but it's been quite difficult to study quiescent neural stem cells in the adult mammalian brain, and this is for several reasons. I've pointed out a couple here. One is that there haven't really been clear markers allowing those stem cells to be identified, and also they tend to be interspersed with cells that are actively dividing. And so we work on a much simpler system than human beings. We work on a Drosophila, the fruit fly, and we think this is a particularly good model system to study these events because Drosophila has neural stem cells which divide in the classic asymmetric fashion to produce neurons in the embryo, and then almost simultaneously they enter this quiescent state. And again, nearly synchronously, they will start dividing again to produce the neurons that are going to make up the adult nervous system. Now, in Drosophila, we can very easily identify these cells. We have markers for them. There's quite a large population, and we're also able to manipulate them and to probe them, find out which genes are on or off, and what stimuli allow them to start proliferating again. But what's quite intriguing is that if you look at these quiescent stem cells in the fruit fly, they actually have a lot of similarities to mammalian neural stem cells. So what I'm showing you on the left here is a diagram of radial glial-like cells in the mammalian brain. This is in the cortex, this is a ventricle here, and what I'd like you to see is that they have these long extensions which head down to the blood-brain barrier, and they also have cilia which project into the lateral ventricle. Now, if you look at a neural stem cell, quiescent neural stem cell in Drosophila, it has many of these same features. So it has these long projections, and this is absolutely characteristic of the quiescent cell. When they're dividing, they're round and proliferating. So this is absolutely characteristic of the cell when it's dormant. So again, they have these long projections, and they have these little end feet. And we believe that these projections are reaching out for a signal that may either tell the stem cells to remain quiescent or perhaps tell them to wake up. We don't know yet. This is one of the questions that we're addressing in the lab. Okay. So they look quite similar, and perhaps they have similar responses to stimuli. So another thing that we know is that when these stem cells reactivate, the first thing that they need to do is to grow. So this is a ventral nerve cord, and the stem cells are in bluey green. And when the animal has just hatched, you can see that they're quite small, about 4 microns in diameter, but they grow to about 10 microns in diameter before they start proliferating again. So the first step is growth. And what we also know is that in order to, before they are able to grow and start proliferating, the animals need to feed. And so this reactivation is absolutely nutrition dependent. And it's dependent upon the animals consuming essential amino acids. So if you feed the animals just on sucrose, but not essential amino acids, the stem cells remain quiescent. And so this is an animal that has what we would call being starved animal, and you can see that the stem cells retain these long projections, which tell us that they are indeed quiescent. Once we feed the animals, they will retract those projections, round up, grow, and start dividing again. So several years ago, we decided to ask which genes were activated or repressed when the stem cells exit quiescence. And a former graduate student, James Chell, did these experiments where he took a nerve cord from an animal where all the stem cells were quiescent and compared it to a nerve cord where they had been reactivated. And we just back then used whole genome microarrays to see which genes were on or off. And there was a very obvious result, which was that we found that the insulin signaling pathway seemed to be on when the cells reactivated. So it looked as if the insulin signaling pathway had to be active in the stem cells themselves in order for them to resume proliferation. Now, the insulin signaling pathway in Drosophila is very much as it is in higher animals, except there's only one insulin receptor, and there are 8 DILPs, which stands for insulin-like peptides. But they when the insulin-like peptides bind to the receptor, they activate the classical pathway, which goes through PI3 kinase and AKT and leads to cell growth and proliferation. Now, we had discovered this looking at whole nerve cords, but what we were then able to do was to knock out the insulin pathway specifically in the stem cells themselves and show that they were then unable to reactivate. And so on the left is the control, and you can see the cells are quite large and they're proliferating. But if we inhibit the insulin pathway in the stem cells, they remain small, they remain quiescent. So that was fine. We knew that insulin signaling was required, but what we didn't know at that point was where is the ligand coming from? And so we went back to our microarray data, and what we found was that one of the insulin-like peptides, DILP6, was very dramatically induced upon feeding. And so this is just hours of developmental time, 0 hours is just hatched, and as they feed, you can see the transcription of DILP6 goes up quite dramatically. And what was very encouraging was that when we looked in the starved animals, we could see no induction. So that suggested that this insulin-like peptide might be the ligand. And so we asked where was it expressed, and what we found was it was expressed in these green cells. Here's the ventral nerve cord, and these green cells are glial cells. And if you take a cross-section through the nerve cord, you can see that the glia sit right on top of the neural stem cells. So they're in the right place at the right time to be secreting insulin-like peptides to reactivate the stem cells. Okay. And I should say that these glia actually form the blood-brain barrier in the fly, and I'll come back to that a little bit later. So what we were fascinated by was that these neural stem cells were reactivated by a local source of insulin. There is systemic insulin in the fly, but that is not sufficient to reactivate the stem cells, perhaps because the insulin-like peptides can't get through the blood-brain barrier. And so there's another source of insulin, which are these glial cells within the brain itself, which produce the insulin-like peptides, and they act as a stem cell niche. Now, there's more and more evidence that local insulin signaling is important, not just in the brain, but there was a very nice paper from the Builder Lab showing that in the intestine, stem cells were also induced to divide by an insulin-like peptide. In this case, it's DILP3, but it's also produced in response to nutrition. So DILP3 is produced by the muscle, and it induces stem cell division in the intestine. And it's possible that a similar thing occurs in mammals, because in mouse, in the small intestine, insulin-like growth factor, IGF-1, is also expressed by muscle and may be involved in inducing stem cell division. Okay. So what we knew at this point was that when the animals fed, amino acids in the diet were sensed by the fat body, which is the liver or adipose tissue of the fly, and acts as a nutritional sensor, which then sends a signal or signals to the brain to tell these glial cells in the blood-brain barrier to produce insulin-like peptides to reactivate the quiescent stem cells, which then retract their extensions, round up, and divide. So what we were interested in was how this signal from the fat body, from the liver, is gone. There we go. Pauline's got her own lab at the Pasteur Institute now in Paris. So just to give you an idea, a reminder of what gap junctions are. Gap junctions are intercellular channels that are able to transmit signals between cells. They're formed of hexamers in one cell, which can join up with hexamers in another to form a channel. And these hexamers are made of proteins called connexins or innexins in Drosophila. And they can passage small molecules such as ions, second messengers, metabolites, amino acids, perhaps RNA, and peptides. So it's a way of cells directly communicating with each other. And we were curious as to what would happen to the neural stem cells if the gap junctions were lost. So you could well imagine that the glia might actually be connected to the stem cells through gap junctions. And if that were the case, then we would have thought that if we knocked out the gap junction proteins, either in the glia or in the stem cells, we should see a phenotype. But what we found was that it was only when we knocked down gap junction proteins in the glia that we saw a phenotype. And it was a very dramatic phenotype. The brains were much, much smaller. So here's the control. Here are the two brain lobes. And when you knock out the gap junctions in the overlying glia, you get very, very small brain. And the reason for this is that we found that when we counted the neural stem cells, they were the right number, but they were not dividing. So they were unable to exit quiescence. There was a reduced number of proliferating cells. Sorry, there we go. A reduced number of proliferating cells. So it seemed that the gap junctions were between the glia themselves and somehow joining up all the glia, rather than from the glia to the stem cells. And what we concluded was that gap junctions are required in the glial cells in the blood-brain barrier to promote neural stem cell exit from quiescence. And what I'm showing you here are the septate junctions between those glial cells, which allow them to form this tight barrier. So again, as you're probably aware, the blood-brain barrier acts as a selective sieve to allow the passage of certain molecules into the brain, but prevent the passage of other molecules. And in mammals, it's formed by endothelial cells in glia, which are joined by tight junctions. Similarly, in Drosophila, there's also a blood-brain barrier, although the blood is actually called hemolymph. And it's formed, the blood-brain barrier is formed totally of glial cells, and they are joined by septate junctions. But again, it acts as a selective sieve to regulate what molecules can enter the brain. And just to show you what that looks like in real life, I've shown you the septate junctions, and this is a 3D form-filling model of the glia. Here are the stem cells in white, and here just showing the glia surrounding the ventral nerve cord and forming this barrier. So I've mentioned that insulin is required to reactivate the stem cells, and I've now said that gap junctions in the glia are required. So the obvious next question was, are gap junctions required in the glial cells to allow insulin secretion? And the answer is yes, indeed, they are. So here I'm showing you a diagram to explain what the image is on the left. So the glial cells are in red, and the insulin-like peptides are in green, and you can see here the secreted molecules, and there may be some retained within the glia. Now, if we starve the animals, I hope you can see that almost all of the insulin is retained within the glial cells, and not very much, if any, is secreted. And similarly, in the gap junction mutant, when we remove the gap junctions, the insulin-like peptides are retained within the glial cells, not very much secreted. So all this time I've been talking about insulin, and I think anybody in the audience, if I mention insulin, would immediately think of the pancreas. And so we decided to ask whether there were any parallels with the pancreas, because of course there have been so many studies on the pancreas and insulin secretion, we thought perhaps we could learn something from prior knowledge there. So this is an artist's impression of the pancreas that was shown at the Royal Society Summer Exhibition. I think it's a very pretty picture. And I just wanted to contrast it with the scientist's view, which is here, which is not quite as beautiful, but perhaps a bit more informative. And what this is showing is a pancreatic beta cell and the steps that occur in the pancreas before insulin is secreted. And so when there's glucose in the bloodstream, it enters into the beta cells and leads to a series of events, including closing of a potassium channel, membrane depolarization, activation of voltage-gated calcium channels. And calcium enters into the cell, and this leads to exocytosis of insulin. So what we decided to look at first was whether we could see any calcium influx in the glial cells before they were secreting or during the secretion of insulin. And these experiments were a little bit tricky because we wanted to do them on live animals, and Pauline Spader did an amazing job just taking these animals and very gently compressing them between glass slides so that they wouldn't move, but still alive. And then we did confocal time series, and we used a genetic marker of calcium, GCAMP, expressed just in the glial cells. And this is what we saw. So this is, we're looking at the glial cells in the brain in green. Well, the calcium is in green. And I hope you can see these beautiful pulses of calcium, these calcium oscillations in the living animal. So these oscillations only occur after the animals have been fed. But what we found when we knocked out the gap junctions was that we could now just see these sort of random spots, random firing, almost like fireworks going across the brain. So we've lost that beautiful coordination, those beautiful calcium waves that we saw, those oscillations that we saw before. And this was really fascinating because there'd been some work done on beta cells in culture, which I'll show you here, showing these lovely calcium waves going across these beta cells. And very intriguingly, when they knock out the gap junctions, they see also this sort of random firing. So what we are seeing in the brain is extremely similar to what's been seen in pancreatic beta cells. And what that said to us was that these gap junctions seem to coordinate these calcium oscillations, which seem to be required for insulin secretion from the glial cells in the brain. Now, it's conceivable that stem cells, neural stem cells in mammals may also be reactivated by insulin signaling. There is some evidence already that insulin-like growth factors can drive proliferation. And also, glial cells, astrocytes, have been shown to secrete insulin-like growth factors in response to injury. So what we wanted to do next then was to see whether we could assess which genes were on or off in response to nutrition. And we wanted to do this in vivo, and we wanted to be able to look at different tissues and organs in their normal environment. So we wanted to assay transcription in the fat body, in the glial cells, and in the neural stem cells themselves. And in order to do this in vivo, we developed a new technology, which we've called targeted DAM-ID. And I'll take you through how this works. What we were aiming to do was to be able to profile genome-wide in a living animal where proteins were binding to the DNA. And I'll explain how we do that now. So this technique is based on a technique called DAM-ID, which was developed by Bas van Steensel and Steve Hennekoff. And it's a way to identify protein binding sites throughout the genome. And it gives you results that are fairly similar to chromatin immunoprecipitation experiments, but it does not require antibodies or cross-linking or cell isolation. So what you do is if you have a DNA binding or chromatin binding protein, you fuse it to an E. coli DAM methylase, make a transgenic animal or do transient transfection, and wherever your protein binds on the DNA, the methylase will methylate on an adenine in the sequence GATC. Now, adenine methylation is extremely rare in vertebrates, and eukaryotes, I should say, and so then all you need to do is to cut with an enzyme that only cuts at the methylated sites, and it will release all the fragments where your protein is bound, and then you can just sequence them, and you know throughout the genome where your protein has bound. Now, the beauty of this technique is that it allows you to drive expression of the fusion protein in any cell type you want in vivo, and then you can profile and get genome-wide data, as I said, without antibodies or cross-linking. And it requires very, very little material. So we've done some experiments reproducing what was done in cells requiring, for chip experiments where they required 6 to 10 million cells, and we've got beautiful results with 10,000. And so we have high hopes for using this in higher animals as well. So I mentioned that we were interested in which genes were on or off, and so the protein that we fused DAM to was RNA polymerase, because that would allow us to see where the polymerase was transcribing in vivo in a cell type specific fashion. And this works, again, very, very beautifully, just by fusing Pol2 to the DAM methylase, and as I said, digesting and getting the fragments out where the polymerase is bound. And I'll show you some data in a second. But the experiment that we did was to look at the neural stem cells and profile transcription in them when they were proliferating, when they became quiescent, and when they were reactivated. And I'm just going to show you one of the genes that has come out of this study, and it's a gene called Tribbles. And what you can see, here's the gene outlined here. When the cells are quiescent, the polymerase is sitting right across the transcription unit, but it's not there, or very, very low levels, before they're quiescent, when they're proliferating, and when they reactivate. So this looked like a very good candidate for being involved in quiescence of neural stem cells. Now, we were able to look at mutants and see what happens when we remove this gene. And what we found is that this gene Tribbles, which encodes a pseudokinase, is required for the stem cells to be quiescent. So in a control animal, when most of the cells are quiescent, you can only see about 4 or 5 cells dividing. But when we remove Tribbles, you can see that there's a far, far larger number of stem cells dividing. And that's quantified here. So here's the control, and you can see the increase when we remove Tribbles. So why is Tribbles required for quiescence? Well, within the last few years, it's been found that Tribbles is able to bind to AKT and block its activity. And this makes perfect sense with what we know about insulin signaling and stem cell reactivation. So it blocks AKT and prevents activation of the insulin signaling pathway. So that seems to be true in Drosophila, but we were also interested to know whether this could be extended to mammalian brains. And there are indeed 3 Tribbles homologs in the mouse, Tribbles 1, 2, and 3. And we wanted to know whether they were expressed in the brain and indeed whether they played a role in the neural stem cells in the mammalian brain. And we've collaborated with Ana Delgado Fumero and Fiona Deutsch, who are at the Biozentrum in Basel. And what we've got very preliminary data which suggests that they are indeed expressed in the stem cells in the adult brain. And we're just characterizing to see whether these are the quiescent or the active stem cells. But in the meantime, what we decided to do was to take these mammalian forms of Tribbles and put them into Drosophila and see whether they could substitute or act in the same way as the Drosophila Tribbles. And we found that indeed they do. So if we express either tribbles 2 or tribbles 3 in flies, you can see that we can cause the cells to become quiescent with an increase in quiescence here. So what that suggested to us, and is very encouraging, is that the function of tribbles seems to be conserved between mouse and Drosophila, and that mouse tribbles can induce quiescence in Drosophila. And so that suggests that perhaps when we look at the mouse brain, we may find that mouse tribbles has a similar function in the mammalian brain in quiescent, neural stem cell quiescence. So for the future, we've been modifying our technique so that we can use it in mammalian cells in an inducible way using Cree and floxed cassettes. We've also adapted the system so that we can map chromatin states, looking at chromatin protein bindings, and also we've made a new technique that allows us to look at RNA chromatin interactions. And we're looking at, as I mentioned, changes in the fat body to see if we can identify the signals to the brain, looking at further changes in the glial cells, and also looking at epigenetic changes in the stem cells themselves. And what this is allowing us to do then is to study communication between different organs in response to nutrition and other stimuli. We're looking at cell-cell signaling between stem cells and their niche, and looking at genome-wide transcriptional and epigenetic changes. And as I mentioned at the beginning, the long-term goal is to be able to understand sufficiently how to wake up dormant stem cells as a means of therapies. And this is something that we maybe look forward to in hopefully not too distant future. And I'd like to just thank the people in my lab. I've mentioned everybody who did the work during my talk. This is the rest of the group. And I'd also like to thank the Wellcome Trust, Herschel Smith Fund, and BBSRC for funding. And thank you for your attention. I guess we don't have a chair, so.
Okay, I found your talk very interesting indeed. I worked on the beta cells some years ago, and one of the interesting things was, as you referred to, that isolated beta cells secrete very little.
Yeah.
Only when they're together and communicating do they work. And you obviously found this also now in the glial cells. However, it is still not clear to me, and I don't know whether maybe it's more clear to you, why it is so important that the calcium signals can be coordinated. I mean, every single cell will have a calcium signal, calcium will stimulate secretion. Right. So why is it actually important that they're coordinated? And we have speculated about that in insulin secretion field, and I'm not sure that there is a good answer to it. And I wonder whether you have found a good answer to it.
Well, I have to confess that this was one of the questions I had, and that's why I went to the pancreas literature, hoping you would be able to tell me what it was. But what I can say is that when we knock out the gap junctions and you see the random firing, if you wait long enough, occasionally 1 or 2 stem cells will reactivate. So whether the coordination has something to do with threshold of insulin signaling, that may be one possibility. What we'd like to do is to knock out insulin signaling in groups of glia to see whether the neural stem cells are rescued by adjacent glia. And that might allow us to get at that question, whether there's a certain what's the smallest number of cells you need in order to get enough insulin?
Okay, Dr. Brand, thank you for the excellent lecture you gave us this afternoon. Okay, Juan de Obarrio from Panama, and I have a question. Okay, we have insulin as a hormone secreted by the pancreas, is that right? And we also have a polypeptide, insulin, a growth factor, insulin-like growth factor.
Yes.
That's totally different than insulin. Yes. Now, my question is the hormone or the polypeptide good for the neural stem cells, or I'm confused about that? Okay.
Well, in Drosophila, we have 8 insulin-like peptides, and they're expressed in different places, but they have a very interesting transcriptional interregulation. So if you knock out one, so for example, DILP6 is the one that seems to be involved in the brain. But if you knock it out, transcription of the other insulin-like peptides is increased. So it does look as if they're able to substitute for each other. So they may not have very different functions the way you've pointed out about insulin and insulin-like growth factor. At least a subset of them are able to function in the same way, but they seem to be expressed in different tissues at different times. Why they're transcriptionally controlled in this unusual way, we have to knock out 4 of them at once in order to actually see an effect, because they will substitute for each other. Does that begin to answer your question? So at least in flies, there seems to be quite a bit of redundancy in terms of their function. So that may be a little bit different from mammals.
Yes. Let's see if I understood. Yeah. That the polypeptide is growth factor, insulin-like growth factor, is good for the stem cells, the neural stem cells. Is that right?
In mammals, IGF-1 or IGF-2, yes. In mammals, yeah.
Thomas von Sklinitsky, UK. Very nice work. I have actually 2 questions. One is, would your data suggest that it should be sufficient to activate calcium influx in the glial cells to induce regeneration? And the second question is, what happens to the gap junctions during fly aging?
Okay. Well, to answer the first question, we haven't done that experiment. We haven't. Yeah, and it should be doable, but I'm afraid we haven't done it. Sorry about that. The second question, I can't.
There must be people who have done experiments like that before they knew about stem cells by calcium.
Yeah.
Modification of calcium channels is done all over the place.
Whether someone's actually done it just in the glial cells in the blood-brain barrier, I doubt, but we can do that now. I'll go back to the lab with that as a suggestion. But in terms of the gap junctions, what I didn't show, which is really intriguing, is that the gap junctions seem to be what we call heteromeric gap junctions. So they're formed of 2 different innexins, innexin 1 and 2, and we see them co-localizing only when the stem cells are being reactivated. So after they've been reactivated, they no longer co-localize. So this suggests there's another level of regulation, which is that at least this type of gap junction is only formed when the stem cells are being reactivated. So that's another area to investigate.
Really great talk. I just had a quick question. If you disrupt the blood-brain barrier, would you induce neurogenesis?
If you disrupt the blood-brain barrier,
nowadays there's a lot of treatments like that where they disrupt their, like maybe for astrocytes for cancer treatment.
I think it would depend at what time in development you did it. So we've been looking at the formation of the blood-brain barrier, and what's interesting is that early on it's actually still quite open, and then it closes over time. And actually we think that part of that closure is a signal from the reactivated stem cells back to the glia. So we think it's important for it to stay rather open to allow the insulin signaling. And once the stem cells have already received the insulin signaling, they tell the glia now to close up. So I think it may depend on when you do it. I think maybe later in development, if you disrupt the blood-brain barrier, that could be a problem. But perhaps earlier in development, it's okay. Well, we would really like to be able, so we've been studying all of this early in development, and we've been looking in the adult to see if there's adult neurogenesis, which would be the time to do this sort of experiment. And unfortunately in Drosophila, there's very, very little division in the adult brain, and it's still not clear to me that there is adult neurogenesis in Drosophila, which is one of the reasons we're collaborating with the Deutsch lab. We may be moving into the mouse, where it might be better to do those sorts of experiments.
Hello. Hi, congratulations for your talk. So I have a question relating to the blocking of the gaps in the stem cells. So it could be possible that it's an alternative connection of the stem cells to the glia? And also, do you know if it is calcium mobilization could be sensitive to glutamate release?
Right. So the first question, we've knocked down by RNAi each of the 8 innexins which form gap junctions in the neural stem cells, and we didn't see a phenotype. But the caveat is RNAi is not always totally, it doesn't necessarily knock out transcription entirely. So I wouldn't say unequivocally that they're not required at some degree in the stem cells, but at least we haven't found a phenotype. So there may be some other connection, or there's a small possibility that maybe there are some other gap junction proteins that might be functioning in some way, but at the moment there's no evidence. We haven't looked at glutamine, so I can't answer that side of the question.
Okay, just one last question, please.
Sorry, I think there was just... Sorry, I can't hear you. Yes, it's possible, but we don't know at this point. Yeah. So, yes.
Sorry. Have you tried to look at the effect of neuromodulators like dopamine in your system to see if that have an effect on your stem cells?
Right. Well, there's evidence in vertebrates that stem cells seem to be receiving signals from neurons. And we've been looking at neurotransmitter receptors to see whether we can see any evidence for that. So far, the only evidence we have is that we've looked at postsynaptic markers in the stem cells, and we can actually see postsynaptic markers at the end of that extension. So we think that we're very hopeful that there will indeed be some signaling from neurons.
Thank you very much.